Hey guys, welcome to Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. Today I am here with a small concept that is the experimental proof for the semi-conservative nature of DNA. So now it falls under your chapter number 6 of molecular basis of inheritance from NCERT textbook for 12th standard of biology. Okay, so now before this replication in short, and then we will proceed with the actual topic for today. So now let's see what is this replication. So term replication, okay, as we know, it is made up of two parts here. The first part is replica. So when we are saying a replica is nothing but a copy. So in case of replication, it is a process in which a copy of DNA is being formed. Okay. So copy of DNA is being formed. Now it is normally people say that when you look same like your mother, okay, they say, okay, you are exact copy of your mother or you are exact replica of your mother. So when we say exact replica, okay, that means you have certain qualities and habits of your mother or your looks of your mother, some features of your mother. That's why they are calling you a copy or the replica of your mother. So in case of your DNA also a similar situation comes. Now as we all know that DNA is made up of two strands, right? So it is made up of two strands. So now let us consider this wire. I have taken a wire and I have twisted it around itself in the same way as DNA winds itself around the another strand. Okay, one strand of DNA would wound itself around the second strand. Okay, so same way I have twisted here. So here in case of replication, what happens is the DNA gets unwound. Okay, and each strand, okay, now we can consider this as strand 1 and strand 2. Each strand is going to act as a template. Okay, so here what happens is this one original strand, they will be called as the parental strand, and the second strand that will be get generated, okay, they will use this original strand from the parental DNA as the templates, and now the second strand will get generated using these templates. So here what happens is the first or the parental strand is still conserved in the daughter DNA. The product is formed as daughter DNA. So still this one strand will be present in the daughter DNA. So that makes it semi-conservative. That means conservative meaning something has been conserved. It is kept intact. But in case of this replication, only one strand of the parent is being conserved. So this makes it semi-conservative. So understand that English term, semi-conservative. Okay. So this particular study or the model was proposed by Watson and Crick when they had postulated the double helix model of DNA. But the experimental proof for the semi-conservative nature was given by Messelson and Stahl, and they had used E. coli as the model organism. Now, what are the perks or what are the advantages of using E. coli? Because everywhere people use E. coli. Its generation time is only 20 minutes. So, in 20 minutes, the progenies would be formed, right? So, it makes the experiment more easy, and secondly, more number of generations can be studied in a short span of time. So this is the advantage. So let's see how did they prove. So if you see this diagram, okay. So as we all know that DNA has two strands, right? One parental strand and one maternal strand. So what they had done was, Messelson and Stahl, they grew this DNA, okay, the E. coli, they grew the E. coli in a medium containing ammonium chloride, and the ammonium chloride contained N15, okay, ammonium chloride. It was the only nitrogenous source that was present in the medium, growth medium that was provided to E. coli. So whatever nitrogen they have to consume, it would be from the ammonium chloride. Fine. And here they use the isotope that was N15. So before we proceed, I want to make it very clear And please keep it in your mind that this N15 is not radioactive in nature. Okay, it is not radioactive, but it is a dense molecule. Okay, and they also use another medium, that is N14, ammonium chloride with N14. So the only difference in N15 and N14 is that they differ in their densities. So ammonium chloride with N15 was a heavier molecule as compared to the N14 containing ammonium chloride. So that they can use the uh, differences in their densities, and we can easily separate out the DNA. So this was the main idea behind using these two different kinds of ammonium chloride which differed in the densities. So after growing the E. coli in a medium containing N15 for around 20 minutes, because the generation time of E. coli is 20 minutes, okay, what they saw was both the strands of DNA contained N15. Okay, so what they had done was they centrifuged this E. coli based on the density gradient, and they saw that all the DNA molecules, they were present in the band that was N15. Okay, so here they saw was it was present. Okay, it was present in both the strands of DNA. Then, after 20 minutes, they changed the medium. They took the E. coli and they transferred them to a medium containing N14 ammonium chloride. So now there was a difference in the density, right? So here we know that in replication, both the strands of DNA, they would separate out, and each one is going to act as a template, right? So in the previous case where N15 was the medium, the newly synthesized strand contained the N15, and both the newly synthesized and the old or the parental strand also had the N15, because for generations they had used that N15. There was no other molecule, but now there was a shift. They had used N14. So, after separation of the strands, the parental strand will have N15. This strand will have N15, right? Both the parental strands will have N15, and because now the new medium, it contains only N14, so now henceforth only N14 will be the source of nitrogen. So the newly synthesized DNA, whatever was produced, it had N14. Okay. So newly synthesized strand of DNA had N14. So we can say that one strand had N15 parental strand and newly synthesized strand had N14. So we can see the difference. So after this, again they transferred the transferred the E. coli to yet another N14 medium. Okay, they allowed them to grow. So now here what happened was, for some time, okay, the initial time, they will have the same model as that of the what we had seen in the second step. That means they had both the parental strand and the newly synthesized strand with N14 and N15. Now again, when the okay, so again now let us see. First step, we will have DNA containing N14 and N15, right? N14 and N15, both the strands are present. So now let us do a replication for this. So now here both the strands are open, right? And henceforth only N14 is present, right? So that means the strand is going to have only N14. So this pink is representing the N14. So here the situation will be that there will be some DNA strands that will contain from the previous one, that is the N14 and N15, and all the later ons or the newly synthesized DNAs, they will have only the N14 containing strands. Okay, both the strands will have N14. That means what has happened was the second strand, whatever was forming, it was the newly one, and still it was retaining the one strand of parental DNA. So this overall it proved the semi-conservative nature of DNA. So here in the predictions they saw was, okay, so it was showing the semi-conservative nature of DNA. Okay, so this is how it was proved that DNA is a it follows the semi-conservative mode of replication. So same study was conducted using the Vishia faba seeds. Okay, that is the faba beans, and this was done by Taylor and his colleagues in 1958. So here they used radioactive thymidine, and they saw that both the strands, they had one parental strand and one the newly synthesized strand. So these experiments, they proved that the DNA in chromosomes, it replicates in a semi-conservative model. So now I was talking about the NEET exams, because here questions could be asked based on this particular diagram. So I request all of you to please go through this particular diagram. It is there in your NCERT textbook also. Try to understand it. So you do not need to understand the whole thing. Okay, you don't need to by heart this. Only thing I will suggest you all is to try to understand this diagram. So if you understand this diagram and if you fit it in your mind during revisions, only it will take some 1 or 2 minutes to recall this diagram. So with this, I will like to stop for today. So till then, keep studying, keep smiling, and if you have any difficulties in your biology, please get connected to us. So like, share and subscribe our channel. Okay, till then, keep studying and keep smiling. Bye-bye.